So many of you here have had a quick peek outside. I can't see anybody obviously just about to arrive, but the reality is that when you do your talk on your home turf, lots of people dribble in late. So I'm sure they're all very busy as well. And it's just lovely to be here this evening for your very special evening, Tihana. You have many people in the room, but also a great many people online and some people who'll be watching on catch up. I'm Jenny Hyam. I'm the Vice Chancellor here at the Georges, and I particularly enjoy inaugural lectures. I just checked with Tihana just to make sure she is going to cover all the bases, and she is going to do from childhood to now, which is exactly what I want to hear. So I don't need to go into too much detail of that, but just to say she has an extraordinary pedigree with preclinical training in Cambridge, clinical training in Oxford, infectious diseases training locally, and as you know, consultant position here since 2013. And along the way, she's done all the right things. She's got the prizes, she's written the papers, she's got prestigious funding, and she's been overall fantastic. So she's going to tell us a bit about a budding mycologist. Little pun, is it deliberately intended there? We'll perhaps hear about that. But I particularly love your vivacity as one of your colleagues. You're one of these people that leaves an impression wherever you go, and I can't wait to hear this lecture. We are going to hear from Tihana. She's threatened me already with the fact she might overrun. We'll see. We afterwards have a reception next door for drinks and canapes. There are no questions in the room, but plenty of opportunity to have a conversation next door. But that won't be until Tom has given the vote of thanks. So without further ado, I'd be very grateful if you come here and deliver your inaugural lecture. Thank you.
Thank you very much, Jenny, and thank you to all my family and friends and colleagues for making the time after a busy day at work, and some who've travelled also quite far. And I think the prize goes to Professor Rodney Phillips there, who travelled from Australia to be here. So it's fantastic to see you all. I'm delighted to be here today, and I'm going to try to cover. People who know me know I like to talk, so I may run slightly over time, but I'm going to try to cover a long period in 50 minutes or so of my journey into medicine, into infectious diseases, how I got into research, including some fairly fortuitous, serendipitous occasions, and also finally how I got into mycology. And there is going to be, you'll see that I've done a variety of things in mycology. I'm not one of these researchers who's done one particular technique my entire career. I've done a variety of things, but the underlying theme and the thread that runs through this is that it's around fighting fungal infection and latterly also tackling fungal resistance. And I'm really grateful to everyone here and people listening online. This has been a journey where I've been supported and encouraged and inspired by many people. If I haven't mentioned you, I apologize, but I have tried to include as many people and events as possible. So just to start, I was born in Arnhem in Holland in May 1972. To my parents, Jasna, my mother who's here, and Tihana Bicanic. Mum has a psychology background and my dad was a civil engineer, and they met doing Yugoslav folk dancing in Zagreb. And 2 years later, I was followed by my brother Nick, and it's as shown here in our house in Sisak, just south of Zagreb. In terms of medical influences in my early childhood, my maternal grandfather, Dragutin Babic, who was born in 1903, studied medicine in Vienna, as you did in those days, And he was a GP during World War II in Bosnia and during German occupation. And he used to go out into the forest at night, risking his life to look after the partisans. Of note, a bit of an infectious disease trivia in the family that I read about in his brother's diary. In the early 1950s, his elder brother contracted TB meningitis in northern Bosnia, and he traveled from Bosnia to Vienna, getting hold of this new injectable drug, streptomycin. But unfortunately, it was a 2-day journey, and by the time he got there, his brother had passed away. I also remember my grandfather's medicine cabinet when I was a little girl. I was one of those children who, is worried about various symptoms, maybe a slight early childhood hypochondriasis, no doubt led me to do medicine. And he always had a pill, a green or red or orange one that would work for my symptoms. Most of my childhood was spent in Yugoslavia, Zagreb, which is now the capital of Croatia. You can see this beautiful, sort of fairly typical Austro-Hungarian era capital. And I lived in one of those typical suburban places, sort of Soviet era high-rise blocks like this one here, Saksova Street. And I went to this primary school, Milo Pavlovich, where we had a really broad education, but there wasn't a lot of there was a lot of memorizing things, and there wasn't a huge amount of creative thought. However, I was fortunate enough in my childhood that through my father's career as an academic civil engineer, we traveled around quite a bit and lived in different countries. So I said I was born in Holland. When I was a 1-month-old baby, they returned to Yugoslavia, and then my father did his PhD in South Wales in 1976 to 8 when I was 4 till 6 years old. And that's when I learned to speak English. We then went back to Yugoslavia, but then he had a sabbatical year at the University of Boulder in Colorado in 1983 to 4. And it was a wonderful time in the Rockies. And as you see, we got to drive this iconic station wagon and pose as a family of cowboys in Colorado Springs. So the next notable thing in the development as an infectious diseases and global health researcher is that I won a scholarship to attend an international school, one of the United World Colleges in South Wales called Atlantic College, built in this idyllic setting around the 12th century St. Donat's Castle. And I'm really pleased that some of my friends from school are here today and also listening online. It was an amazing sixth form college, mixed boys and girls, founded by the educationalist Kurt Hahn. It was all about international understanding and promoting peace. And we were 51 different nationalities. And I'm sure it is one of the reasons that led me to have a passion about global health. And also online, I'm really pleased that Jan Harbour, who was my biology teacher for my international baccalaureate higher level, is also listening. And she was very much loved by the students as a really enthusiastic biology teacher. I still remember her trying to get us through the Krebs cycle for biology higher level. And she also was a leader of the community service I took part in, which was the environmental monitoring unit, where we did various environmental ecology projects, but also learned to scuba dive. I then went on to university at Cambridge, the city of dreaming spires. My college wasn't the sort of typical dreaming spires. You can see them here in the background. It was the brutalist architecture example, Churchill College, which was actually a lovely and friendly college full of engineers. Medics were less common. I have a friend attending, Fatosh here from Cambridge, and we had a really fun time combining laboratory work and practicals. I knew from very early on when I cut through the frog sciatic nerve in a physiology practical where my then partner, John Collier, who's now a MaxVac surgeon, said, T, don't be a surgeon. And I said, don't worry, I won't. I also enjoyed some rowing on the river, and I can't really profess to any blues or sporting successes. I have 2 wooden spoons to my name, which essentially means being bumped down 4 places in the May Bumps races. In my 3rd year at Cambridge, I did, we all did intercalated BScs, but I did something called Anatomy Option B. This had very little to do with anatomy and was a course set up by an anatomy lecturer around the HIV/AIDS epidemic and virus, which was just being described. And we learnt about the virus and about the disease, but also about the societal impact of the HIV pandemic. And my dad said to me at the time, "You're going to end up working with AIDS patients." And I said, "Oh no, come on, Dad, that's not true." But he was absolutely right. And I then went on to clinical school at John Radcliffe Hospital in Oxford. They were a happy 3 years. We learned some really excellent clinical medicine. And this is a yearbook page, and you can see here in red circles are Shamim and Kate, who are both actually consultant colleagues at St George's, and Kate is a fellow professor at the university. Simon Morley here, who I hope is a bit cleaner now, but wanted me to share this fungal photo of his student fridge, which he sent me for my presentation. I graduated from Oxford in June 1996, here seen with my family. But during my time in Oxford, of course, we all had the opportunity to go on electives to exotic places, and they were 10 weeks at the time, as long as we could find sponsorship and funding. So mine was spent, and this was my first trip ever to Africa and South Africa. I went to the Red Cross Children's Hospital, a paediatric hospital in Cape Town, followed by Edendale Hospital in KwaZulu-Natal, where I encountered, I was exposed to medicine in a more resource-limited setting, and I saw my first examples of both paediatric malnutrition and various infectious diseases I hadn't encountered to date. And it's when I definitely fell in love with Cape Town and South Africa for the first time. I also met on a hike to Drakensberg some people who were actually, I encountered again in Cape Town 10 years later, and who were to become good friends. I then did what a lot of my generation at the time managed to arrange. So after house jobs in Oxford and Glasgow and an A&E job at St Thomas's, I then went to Australia for a year as a senior house officer at Flinders Medical Centre, which was a teaching hospital in Adelaide. I lived in this flat with a glorious view of the sea, and it definitely wasn't all work. And I got my first taste for wine tasting, which I definitely continued in South Africa, and got to travel in the outback as well as on the Great Barrier Reef. I then returned, did a year as an SHO in Nottingham City Hospital, and passed my membership exam. And I wasn't 100% sure what I wanted to do as a specialty. So I thought, okay, well, why don't I do an ICU job in London? So I applied for a job at St Thomas's, which, and this is one of these serendipitous events in one's career, when a job came up that was 6 months microbiology, 6 months intensive care. I applied and was interviewed by the formidable Susanna Eakin, who was a great clinical microbiologist at the time, and trained many microbiologists, including some of my colleagues in the room today. And certainly all surgeons feared her because she used to summon them to her office if they had unusual post-operative infection rates. I was taught my first microbiology by Bruce McRae, also South African, who's here in the audience. He taught me, as he taught medical students, that bacteria divided into pinks and purples and cocci and rods. And he also, on a notable weekend on call, in those days, we were a bit less conscious of safety. So microbiologists used to sniff plates, and there was a little technique, a bit like wine tasting, where you wafted it under your nose and inhaled. Anyway, Bruce had shown me what he said was a classic example of Neisseria gonorrhoeae, which later turned out to be Neisseria meningitidis, the causative agent of meningococcus. So Bruce and I promptly took ourselves off to A&E at St Thomas's and got some ciprofloxacin. I also published my first case report. A note for any students who are here, you know, when they say you've got to start your academic career young, well, I certainly, this was my first ever publication and I was already an SHO. I also met Duncan Winkle, who's intensive care consultant. It was a great experience for me doing intensive care, and he was just a young consultant then, and then again, another serendipitous event bumping into him about 20 years later, we set up some ICU studies together, cohort studies. I then I was really keen to get some infectious diseases experience in the tropics under my belt, and I had been interviewed for and awarded an infectious diseases and microbiology training position here at George's, but I was allowed to start with a 3-month delay, and I went out to Thailand to Professor Nick White and also Aryan Dondorp, notable malaria and other tropical infectious diseases researchers at a Wellcome Trust facility in Bangkok in Thailand. I was sent out with a sort of overnight in Bangkok to northeast Thailand, Sapasit Pasong Hospital in Ubon Ratchathani, where together with a Thai nurse who started the same day as I did, and after a one-day handover, not really any good clinical practice training at that time, I was enrolling through my Thai nurse patients into my first ever randomized clinical trial, comparing ceftazidime or ceftazidime combined with cotrimoxazole for this infection, bacterial infection called melioidosis, which is a bacterium that's present in the soil. It's exposed, particularly rice farmers are at risk during rainy season when it's either inoculated through the skin or inhaled, and causes very nasty sepsis as well as lung and liver and spleen abscesses. As you can see here, this paper led to a publication, but as with all these massive endeavors involving lots of people, my name did appear just in the acknowledgements. I also discovered a love of Thai food, and indeed Nick White did say to me at the time, why do you want to go to Africa? The food is much better in Asia. I then came back and started my specialist training in infectious disease and microbiology, and myself and Tim Planch, who's here in the audience, were the first 2 joint trainees to be appointed by Mark and George, who's sitting here in the audience. And you can see here, it was quite a male-dominated department. This was the infectious diseases consultants, 5 of them at the time. I was one of the first, if not the first female registrar, although Mark once remarked it felt like 5. And we were here on Mackenzie Ward, and there was actually a really lovely legacy of infectious disease at St George's, where previously there was a fever hospital set up in the late 19th century called the Grove Fever Hospital for victims of scarlet fever, diphtheria and enteric fever. And Charles McEntee was a superintendent there, and Mackenzie Ward, which is our dedicated infectious diseases ward, is named after him. And you can see here we were there's Sister Linda and Sister Marjorie, who may be joining today from Alaska. They were fantastic ward sisters. And you can see I got to work with people who all went on to careers in infection. John Clark, who's a microbiologist at St Helier, Charlie Woodrow, who went to Thailand, and Joe Jarvis, of course, who you'll hear more about later. And ably supported by Wanda, our departmental mama and secretary. Wanda has known me such a long time that when she saw me today, she was able to say, "You look like a British Airways hostess." Just for Wanda, where are you? Wanda, I just want to point out that the emergency exits are Okay. So, of course, it's not such a male-dominated team nowadays, and I'm really fortunate to work in a fantastic team of, this is my clinical team. We've merged as a joint infection department with microbiology and our HIV colleagues, and many of them are pictured here. And as you can see, we have a good spread of both age and gender across the department. And it really is a pleasure coming to work and working with such a great team. Again, in those days, I slightly feel for trainees nowadays, because training is much more rigid. And in those days, there really was a lot of flexibility. And I was very grateful that as part of my MSc in microbiology, I managed to negotiate a 3-month trip to Gabon in Central Africa, where I was doing a project supervised by Sanjeev Krishna and supported greatly by Tim Planche. And Joe Jarvis actually preceded me in Gabon. He was there for 4 months and I was there for 2 months, finishing off a study. We did a study about dehydration in children with severe malaria. So again, this was a real jumping in at the deep end. I had to take blood from 2-year-olds. I had to talk to the mothers in French. I had to do a crash course in French before going out there. But it was all tremendous fun and a success, and even had my hair plaited African style, and also had some quirky characters, including the French medical superintendent who used to attend the hospital mess dinners accompanied by his pet chimpanzee. And this led to a publication in Clinical Infectious Diseases. But what about mycology? I said I was going to tell you about my career in mycology and introduce you to some fungi and fungals and antifungals. So just as a background to those who don't know anything about mycology or medicine, I introduce to you the big 3 killers in fungal infection. We have Candida, we have Aspergillus, and we have Cryptococcus. I've deliberately made Cryptococcus the hippo because that kills more humans in Africa than any other disease. Candida is a gut commensal that is present in all of us, but overgrows in the setting of antibiotics and can cause things like oral thrush, and also can stick to lines and plastics and is associated with line infection in patients, particularly on intensive care, where it can enter the bloodstream and cause sepsis. Aspergillus is a mold that's inhaled from the environment and causes lung infection, particularly in people with structurally abnormal lungs, which can be nodules or become severely invasive in the setting of things like leukemia and chemotherapy. And then Cryptococcus causes this brain infection known as a meningoencephalitis. And it's this yeast shown here in an India ink stain, a yeast surrounded by a halo, and that halo designates its protective polysaccharide capsule, which protects it from immune killing. Now, fungi are conventionally, all of us mycologists like to bang this drum, but it's true that fungal diseases are far less heard of than bacterial diseases, and they are neglected, but they do actually affect a lot of people globally. So these big 3 collectively affect over 1.6 million infections, cause every year, with mortality rates depending on treatments and availabilities up to, as you can see, 90%. Why? Also because they often affect hosts who have compromised immune systems. In other words, whether it's through what doctors, the medicines that they give them, or being on intensive care, they are opportunistic pathogens, and they affect people and lead to a high mortality rate. So fungi are definitely increasing as a cause of human disease with advances in both chemotherapy, organ transplantation, the HIV epidemic, intensive care, where more sicker patients with multiple morbidities go on to ICU for long stays, the development of biologic treatments for things like rheumatoid arthritis and other inflammatory diseases, and also probably climate change, because fungi by nature are thermotolerant. They can withstand higher temperatures, and they are environmental organisms. So what about antifungal drug development? Well, this is the timeline as shown here. So amphotericin B, still a very widely used antifungal, was developed in the 50s. The problem with it is it's still only available in intravenous form and can be toxic to the kidneys. The next major class to be developed in the 80s and 90s were the azoles, which are available in tablet form and are most widely used antifungal class. And then more recently, there's the echinocandins, again, which need to be given intravenously. And the vast majority of fungal infections that we treat are treated with one of those three classes, or sometimes a combination of, unlike antibiotics, where we have a much broader arsenal. But the fungi, a bit like bacteria, adapt quickly and they become resistant. And this is becoming an increasing issue for these three drug classes, as you will see later on in this talk. And even we have some new antifungals emerging, and there's a capacity to become resistant to those. So as a result of emerging resistance and a global disease burden and the high mortality, the WHO published last year their fungal priority pathogens list. And you can see here that the big three, Candida, Aspergillus and Cryptococcus, are critical priority because of their ability to cause disease, but because also resistance is emerging in all those fungi. And in particular, there's a new kid on the block that's much feared called Candida auris, which is a fungal superbug. It's like the fungal MRSA. It can become resistant to multiple, in fact, all three drug classes, and is usually always resistant to the azole drug class. So about my career as a budding mycologist, well, again, a chance thing. So Tom Harrison, who's going to give the vote of thanks later, said to me about my second year here at St. George's, how would you like to do a project in Cape Town? I had a year allocated in my training to research. I said, I'd love to go to Cape Town. What's the project? And he said, it's on cryptococcal meningitis. I had seen a few patients on McKenty with this disease, and I knew it was a deadly brain infection. And even before I went, Tom got me to write a review and read hundreds of papers to write probably what is still our most cited review paper in the British Medical Bulletin. Essentially, this disease, like a lot of fungal infections, is in the environment, the Cryptococcus, in spore form, associated or dried yeast form with either pigeon poo or tree bark, and some bushes, also the mapana tree in southern Africa. It's inhaled and usually contained at the level of the lungs by innate immune cells called macrophages that gobble up the Cryptococcus and then present it to other immune cells that come in. They're called T cells, and they help to kill this pathogen. However, in the setting where your T cells are not working, or you don't have any T helper cells, such as HIV/AIDS, this can enter the bloodstream and disseminate to the brain, causing this very nasty brain infection characterized by weeks of severe headache, sometimes accompanied by coma or drowsiness, confusion, seizures, and also pressure on some of the key nerves, including the optic nerve causing blindness and the auditory nerve causing hearing loss. And it's diagnosed by doing a lumbar puncture, which is essentially taking a tap of spinal fluid. And you can see here I'm using a manometer to measure the pressure of fluid that comes out, because this is a disease that often is accompanied by so-called raised intracranial pressure, which can have a detrimental impact on both hearing and vision. And it was diagnosed at that time. I've already shown you a picture of this black, literally writing ink, India ink, with this halo, of course, showing these encapsulated yeast. It's a very significant infection, and it's still the most common cause of meningitis in Africa. It's a so-called AIDS-defining infection. When you have cryptococcal meningitis, it means you've reached a severe stage of immunosuppression. And it's been shown in studies across the continent that about 1 in 16 patients with AIDS have this bug in their bloodstream, a sort of pre-meningitis stage. And it causes about 180,000 deaths a year globally, of which 3/4 are in Africa, largely because of the HIV pandemic. So I was very fortunate that Tom sent me out to South Africa on a project. It was at an amazingly timely, opportune time to really make an impact, because it was at a time of AIDS denialism in South Africa. Remember that here in the UK already in 1998-99, we had rolled out antiretroviral therapy and our HIV patients were doing quite well. I had arrived in January 2005. I arrived in September, and the public sector antiretroviral therapy rollout in South Africa started in the January. So at this time, working as in a township hospital, we were seeing about 3 cases of cryptococcal meningitis every single week. And in fact, I just looked up the adult HIV prevalence in South Africa is 17%, then it was 11%. But if you look at the antenatal pregnant women population, it's more like over 30%. So these were the young patients that we were seeing. And I was working with Linda Gale Becker and Robin Wood at the Desmond Tutu HIV Centre, but the vast majority of my work was at a township hospital with an infectious disease amazing service led by the inspiring Graham Menkes and Kevin Reeby, and supported also by Mark Mendelsohn, shown here. And this is the hospital, and you can see it's on the edge of Cape Town's biggest township, Khayelitsha. It was in Manenberg. And we were working on these open plan wards, no isolation or very few side rooms. And you can see here, here's my nurse, Bondi, and she was recruited to help me and translate into Xhosa. And here she is administering amphotericin B, this intravenous medication, to one of our patients. Now, again, to just give you an idea of the setting at the time, the US guidelines at the time, based on US trials, recommended a combination of amphotericin B and flucytosine was the best gold standard treatment for cryptococcal meningitis up front, stepping down to the tablet fluconazole that I mentioned to you was part of this azole drug class. However, in much of sub-Saharan Africa, fluconazole was not, amphotericin B nor flucytosine was available. And the only drug available was diflucan, which was the trade name for fluconazole, provided by Pfizer free of charge in the continent. So we didn't have amphotericin B available, or if it was available, the supply was intermittent and it was expensive. Now, again, as luck or bad luck would have it, when I got there, I was intending to, we had a protocol for a randomized trial comparing 2 different doses of amphotericin B. However, I'd meant to go there for a year. It took 9 months of bureaucratic wrangling to get the relevant permissions to do this trial from the South African Medicines Control Council. And Graham and colleagues, seeing my frustration, they let me do some clinics. But Graham said, look, I've got this bunch of notes from patients that I collected who are relapsing with fluconazole resistance, and we're having to treat them for 6 weeks with amphotericin B. Why don't you just audit and analyze this? And this was my very first author publication, where we essentially showed that in the setting of fluconazole monotherapy, fluconazole resistance, secondary resistance, which you can see here is in the red zone here. So about 2/3 of cases of relapse were caused by drug resistance. And this had a high mortality of over 50%. And this was, like I said, in the context where antiretrovirals to restore the immune system were not available, or there was a delay, 5 months delay. So a lot of these patients were dying of a very difficult or untreatable infection. Again, whilst waiting to start my real study, I did what actually turned out to be probably my most impactful study, which was an observational study that Tom suggested that I do whilst waiting. He said, why don't you, when amphotericin B is available, and when it's not, why don't you do the same study we intended to do? And this is something called monitoring the rate of clearance of infection. So these graphs here essentially involved me doing a lumbar puncture and taking the spinal fluid from the patient at diagnosis before treatment, and then twice during the first week, and then again in the second week. So multiple lumbar puncture. I would then transport the fluid to the lab, which was about 2 minutes away. It was a wonderfully small, friendly hospital, where I would then culture it and count the number of colonies per milliliter of CSF. And I would plot a graph, and each individual patient had this graph of how they cleared the infection. And it's not difficult to see that the rate of clearance of infection for amphotericin B, a fungicidal agent, that it kills the fungus, and many people sterilize their spinal fluid, clear the infection completely by 2 weeks. Whereas for fluconazole, it is essentially fungistatic. That means it controls the growth of the fungus. It doesn't go up, but it certainly doesn't clear it. And this was a really significant finding at the time, which we published. And at the same time, one of the other barriers to widespread use of amphotericin B in the public sector in South Africa is I discovered when trying to procure it for my trial that the price that the South African government had somehow agreed to with Bristol Myers Squibb was around 12 or 13 pounds a vial, whereas the rest of the African continent were only being charged £2 a vial. So I contacted a pharmacy colleague at the local Médecins Sans Frontières, as well as a powerful advocacy group called the Treatment Action Campaign, who had been lobbying the government for access to antiretrovirals. They commissioned a lawyer who threatened to take the company to the competitions court, and promptly we got them to reduce the price to around £2 a vial, thus enabling both the research, the efficacy I'd shown, as well as the price, enabling this drug to be actually rolled out across the whole of South Africa. And this was very exciting, and I would say probably the most impactful thing I'm ever likely to do again. And it's one of the things that makes research, clinical research, so gratifying. Our group then went on to publish more articles and start an effort led by my colleague Angela Loys for advocacy for access to medicines and also diagnostics across sub-Saharan Africa for cryptococcal meningitis. This is the randomized trial I went out to do, which eventually 9 months later I started doing, and it took another year. And this compares 2 different doses of amphotericin B. And again, you know, with the eye of faith, you can probably see that the higher dose, together with another drug called flucytosine, is even faster at clearing infection. And most importantly, as I mentioned, it's toxic to the kidneys. This was achieved, we managed to manage the kidney toxicity by giving people careful hydration and management of electrolytes. So, as similar to the view of Signal Hill from my balcony here in Cape Town, we managed to show, essentially this is my cohort, recruited over 2 years, this is how common this infection was, of almost 120 patients, that the patients who survived the early period and were started on antiretroviral therapy had a good long-term outlook if you had access to the amphotericin B and antiretrovirals to restore the immune system. So this led to an update of the South African guidelines in 2006, as indeed, as my wise friend Chris Whitty said to me before I went out to South Africa, research buys you a place at the policy table, and how right he was. And myself, but also my research team, have been really fortunate that our work has led to, this later on led to WHO guidelines update. And even just this year, Tom and I have been involved in a huge project to update the global guidelines for cryptococcal meningitis. Meanwhile, the team carried on and went into so-called phase 3, much larger trials where mortality was an endpoint. This is Sheila Malloy from our group, and Sheila and Tom and colleagues from 4 African countries did this huge trial, the biggest ever to that time, cryptococcal meningitis trial, to show again that amphotericin B and flucytosine had the best survival, 10-week survival, as well as the optimal fungal clearance. So again, this was translated into a change in WHO policy. And then lastly, Joe Jarvis, who after St. George's went to the London School together with Tom, did a recent trial called the Ambition trial to show that just a single dose, high dose of amphotericin B in a liposomal, which is a less toxic to the kidneys formulation, was just as effective in terms of mortality as the standard of care regimen of amphotericin B and flucytosine when given on a backbone also of fluconazole and flucytosine. So, and this has been the biggest trial done to date of cryptococcal meningitis, again leading to a WHO guideline update. Now, what of the complications of the disease? So while I was in South Africa, I noted, obviously, when you see over 100 cases of a single disease, you're in a great position to describe some of its manifestations and complications. So one of the major things that I mentioned to you is that this organism, as it's dying and shedding capsule, can obstruct outflow of spinal fluid from around the brain through these structures called the arachnoid granulations and through the lymphatics. And a normal opening pressure, when you do a lumbar puncture and you look at the level of the fluid, it should be less than 20 centimetre. Anything above that is defined as raised intracranial pressure. And up to 3/4 of patients with cryptococcal meningitis have this problem. By looking at a cohort, my own cohort from Cape Town, as well as a prior study we did in Thailand, I was able to show that the amount of fungal burden that you have in your spinal fluid at diagnosis is related to your likelihood of having raised opening pressure. However, the relationship was not linear. If you had high pressure, you generally had high fungal burden, as shown in this graph here. But at the lower levels of pressure, there was a whole range. So perhaps there could be some other factor causing it. And you can see here in a post-mortem study that we published of these filter structures of the brain, the arachnoid granulations, you can see these multiple yeasts that are, if you like, trapped in there. And you can see on this EM, this electron micrograph, these big fat capsules around the organism. And I mentioned to you that the capsule is something that the organism produces to defend itself from the immune system. And different strains can produce different levels of capsules. So this gave an opportunity to do some preliminary pilot work for my Wellcome Intermediate application, which I went to New York, Albert Einstein College of Medicine for 3 months and worked with Bettina Fries and Arturo Casadevall and colleagues, looking at essentially, I took all my clinical isolates from Cape Town along and I was profiling them according to capsular size and shedding, and I learned how to do that. And they had shown that if you infect a rat with different variants of Cryptococcus, so-called smooth and the mucoid variant, which produces larger, stickier capsules, the rat is much more likely to develop raised intracranial pressure when infected with the mucoid variant. So to test whether this association was true, of course, animal models are much more controlled settings, but I thought, again, let's do an ex vivo study. Let's do a human study. So I was fortunate that David Bulwer, my friend and colleague from University of Minnesota, was starting a trial in Uganda, in Kampala, Mulago Hospital, as shown here. And he allowed me to do a sub-study of this capsular phenotype in patients in his COAT trial. And he literally threw me in at the deep end, both literally on this whitewater rafting. I've never been so terrified in the Nile, but also starting a new study and helping him to get the lab work sorted in the microbiology lab. And this is my postdoc, Emma, who did the lab work back here, and Grace Najuka, who really should have worn glasses by the end of this, because she literally did India ink staining and measured the capsule in hundreds of cells from hundreds of patients and looked at whether the size of the diameter of the capsule in these patients' CSF related to pressure. And indeed, she did find that at very high level of pressures, the patients had higher, bigger capsules. And we also found that the capsule, the bigger the capsule, the more likely that people would not show an inflammatory response. So the capsule dampened down inflammation in patients. Later on, together with Sheila Malloy, we also analysed the relationship between the fungal burden, the raised pressure, and visual loss. And we found that visual loss, from just some visual loss to blindness, was associated with these complications. Hence, again, the importance in treatment guidelines of managing this aggressively by doing therapeutic lumbar punctures, where you literally just tap off 20 to 40 milliliters of spinal fluid each time. We also, Wilbur Sabiti in my group, who came from Robin May's group in Birmingham, they were working at the time, they had a lovely in vitro cell culture model using mouse macrophages, where they looked at how effectively, so I mentioned to you that the lung macrophages are the ones that gobble up the cryptococcus and contain it. However, the cryptococcus, as Robin's group and others showed, was able to actually replicate and survive inside macrophages and burst and actually spread to other macrophages. Not only that, but the clever fungi used the macrophage as a sort of Trojan horse. They hid within the macrophage, and that's how sometimes they got into the brain unchecked by the immune system. So we took some isolates from my patients in Cape Town for whom we knew what the fungal burden was and their survival, and we looked at how effectively they were taken up by macrophages. And we were able to show, in this study, Wilbur was able to show that the ones that were highly taken up were much more, were associated with a higher fungal burden and also a worse survival. Matt Beale, who did a lot of sequencing and genotyping of the same isolates, also showed that certain genotypes were associated with worse survival. Wilbur also tested another virulence factor, that is a factor that Cryptococcus uses to defend itself from the host. It's the production of melanin with the enzyme lacase, this black pigment. And again, melanin helps fungal cells avoid the immune system and also fungal killing. And amazingly, we were able to show that the amount of melanin a strain produced in the lab affected how it was cleared in the patient by the amphotericin B treatment. Meanwhile, I had become a consultant, and I was fortunate that Mark Wansborough-Jones retired just at the right time, and I was able to be appointed to his job as a senior lecturer. And I thought, let me take my antifungal interest to the wards here at St George's. And it was the time when stewardship was first, and antimicrobial resistance was becoming more of an issue. And Laura Whitney, who's here somewhere with me, pharmacist, set up the very first antifungal stewardship program, probably, we think in the UK. And we started doing weekly ward rounds and reviewing adult patients on an antifungal. And we quickly realized that fungal prescribing was not just so-called targeted. In other words, in patients in whom they had a known fungal infection, there was a lot of just, we think it might be fungal, so-called empiric. And we were able to show that in the vast majority of cases, as shown in orange here, these patients did not turn out to have fungal infection. In other words, there was a lot of inappropriate prescribing. We hope we managed to talk to teams and stop quite a lot of this. And we also managed to reduce the cost, the spend, from 1 million a year at George's on antifungals down to about 670,000. Then it sort of crept up, and note, this is the year I went on maternity leave to have Sasha. But I still like to wager that we did better than the rest of English trusts, where spend actually doubled. Meanwhile, of course, we were doing all this laboratory work with the cryptococcal isolates, but I was very keen to get back into the real world and get closer to the patient. And in Africa, despite all this work showing amphotericin B was a much better drug, nevertheless, the vast majority of patients were still being treated with fluconazole alone. And again, I mentioned the word empirical. Often they didn't get a lumbar puncture, they got some antibiotics, then they got some TB treatment if that didn't work. And then if that didn't work, they got fluconazole to cover them for all the possible causes of meningitis. And this is Dar es Salaam, where my very first PhD student, Neil, who's here somewhere in the audience, hello Neil, went out to Dar es Salaam to do this study at the National Institute of Medical Research with Sayoki and Fananga. And Neil did a similar thing to what I did in Cape Town. Again, he recruited patients with cryptococcal meningitis into these two cohorts. One was receiving fluconazole at slightly higher doses than my patients in Cape Town, which was the local protocol. And the other group were being randomized and recruited into Tom's big ACTA clinical trial, and they were getting combination therapy with two drugs. Neil did serial lumbar punctures on these patients, but in addition to plating the spinal fluid onto normal agar, he would also plate it onto agar containing the drug fluconazole to automatically find any subpopulations of the cryptococcus that were able to grow on fluconazole. And this is something called heteroresistance. And Neil was essentially able to show in this cohort of around 20 or so patients that you see this black line here is going down of the reduction in the total fungal population over time. But this red line here is this subpopulation that can grow through the fluconazole. And by 2 weeks, the two lines are touching. In other words, the whole population is become resistant as early as that. And remember back to that earlier work we did on fluconazole resistant relapses, and that's exactly what happened in Neil's cohort. Some patients relapsed later with resistance, whereas in the combination therapy arm, that red line goes down to 0, and the combination of fluconazole and flucytosine, shown later in Tom's ACTA trial to be the best oral combination, was the most effective. We also did whole genome sequencing and various other studies with Judy Berman in Tel Aviv to show that the mechanism, the predominant mechanism of resistance was duplication of one of the cryptococcal chromosomes, chromosome 1, on which sits both the drug target, the agostrol gene, ERG11, as well as the pump, the drug efflux pump that pumps the drug out of the cell. We, together with my long-standing collaborator, William Hope in Liverpool, we were able to replicate this in an animal model, in a mouse model, where again, this time, of course, with the mice were sacrificed at regular intervals, and their brains at day 7 were cultured, and the fungal burden was looked at over time. And again, we were able to show that this duplication of chromosome 1 is occurring in mice exposed to fluconazole, the same as it had been in human infection. Meanwhile, in my personal life, I met John, who's an ex-army tank commander, some would argue of a strong enough character and personality to cope with me, and my forceful personality. And we got married in Wadham College, my Oxford College, in 2016. John is not only a leader and a strong personality by nature, but has been a very wise counsel, having himself also experienced after his army career neurosciences research, and he's given me a huge amount of support and encouragement and wise words for my day-to-day career and research issues. The same year, our daughter Sasha, sitting here in the front row, beautiful daughter, was born, and she's now 8 and hopefully still paying attention to this presentation. Then in 2020, of course, we were hit by COVID-19. And given my experience in clinical research, I said to Tom, we've got to do something. Fortunately, the recovery trial, the Oxford group had a protocol ready to go. We stepped forward. We were the principal investigators just for St George's. There were over 180 hospitals, but we recruited, and I can see Tinashe there and Kamal. They were brilliant colleagues that I met through the clinical research facility. And isn't that amazing when research can pull together like that quickly? And from the first patient recruited in early April to June, when we had our first results showing that steroids saved lives in COVID-19, and this could be immediately implemented worldwide. And it was fantastic to be a tiny part of and contributor to that result. At the same time, Johnny Youngs and then later Will Hurt, we'd set up a study with my colleague Duncan Winkle at in the ICU, the intensive care unit, of this mold infection, lung mold infection, aspergillosis, in severe viral pneumonia. And we'd started off, we called it Aspiflu, because it was going to be all about secondary infection in ventilator patients with influenza. However, we pivoted to COVID-19, as did all research, and also included a much larger group of 266 COVID patients enrolled in 5 UK intensive care units. And we found a varied prevalence from 4% in patients, control patients with pneumonia, 11 and 15% of secondary pulmonary aspergillosis, which was a risk factor for death when adjusting for other factors. The risk factors for developing this complication was found in a paper published this year by Will in Thorax. Illness severity, Apache score on ICU admission, and the use of immunosuppressive treatments. Remember, I said that fungi are opportunists. So if you'd had steroids or the IL-6 inhibitor tocilizumab, you were more likely to get secondary Aspergillus infection. And also if you had underlying lung disease, COPD. And Will is carrying on again with some of these patient samples and now doing a PhD at University of Exeter to further dissect the mechanisms of this relationship. And lastly, my current study, CandiRes. So this is an ongoing study, again, coming back to the theme of fungal resistance, which is now an increasing global problem, particularly for Candida, our gut commensal, and as I mentioned, a common problem, particularly in patients on intensive care. We have been recruiting since 2022 adult patients in our intensive care who have various risk factors for Candida infections, and then following them for 30 days and looking at whether an antifungal was prescribed or not and for how long. And we're looking at the proportion of, we're collecting their swabs from their mouth and perianal area, which are then taken to the lab, cultured on fungal selective media. The Candida species are identified and we do resistance profiling on them. And I'm just going to show you one slide of our initial results from a cohort now of over 300 patients. And you can see here, we're again coming back to this drug fluconazole, which a bit like in Cryptococcus is fungistatic in Candida and is the one that's associated with the biggest resistance problem. On the left, we have patients who didn't have any antifungal exposure. On the y-axis, you've got increasing resistance to fluconazole, the so-called MIC, minimum inhibitory concentration. And you have days in the study or days from start of drug. And on the right, you can see that patients who had even 1 day of azoles were much more likely to develop a more resistant isolate over the course of their ICU stay than those who did not. We also further took these studies back in the lab, and again coming back to this concept of resistant subpopulations, which are hiding as 1% of that population of fungi infecting you even before any treatment. And Ben and Claire, who started this, have been doing all this profiling because we have this rich data set of serial isolates. And for example, in this one patient who had a Candida glabrata, which is semi-resistant to fluconazole, over time, you can see this purple population here is able to grow above the break point. And over time, this population gets bigger as the patient stays longer on fluconazole. And eventually, the entire population is resistant. And when we sequence those isolates, we find, again, some genetic mutation in both the efflux pump as well as the drug target. So again, showing the mechanism of how resistance evolves. We are going to, lucky enough that led by Elaine Bignell from Exeter and also partnered again with William Hope, we've now been awarded a 5-year MRC program grant to work further on tackling this problem of antifungal resistance in Candida. And can we use combinations or different strategies and profiling a whole range, screening a huge library of existing and upcoming drugs against different Candida species fed in from the Candires patients, and then test the most promising combinations in animal models and ultimately also move those into human trials. And of course, again, I come back to my first love, South Africa, as a place where we really want to be doing these studies. Why? Because again, that's where the disease burden is highest. And together with Nilesh Govender and my colleague Ray Wake, we are hoping to devise a study of candidemia, Candida bloodstream infection, and combination therapy. In South Africa, unfortunately, especially as you can see here in Gauteng and around Johannesburg, there's a huge problem with this multi-drug resistant Candida auris, which is essentially endemic on the ICUs. And it's coming to the UK, by the way. It's just a bit delayed, and it's already in Southern Europe. In South Africa, the top 2 causes of bloodstream infections are Candida parasilosis, of which 2/3 already are caused by a fluconazole-resistant clone, and Candida auris, which is almost universally fluconazole-resistant. So we hope now to be building the tools and the cohorts and working with UK networks, hopefully building European networks and also South African networks. We are moving towards identifying the best resistant measures to incorporate into clinical trials and markers of treatment response. And we hope to, in the next few years, get funding to do a trial of, again, combination therapy, but this time for treating Candida bloodstream infection. And with that, I just want to thank, none of this research would be possible without multiple funders, which I've been fortunate enough have supported me over years, including St. George's Hospital Medical Charity, as well as industry funders and government funders. But again, none of this, I must stress, wouldn't have been possible without my parents, who've always supported and encouraged me, and in particular my dad, who was a Regis Professor of Civil Engineering and is no longer with us, but I know would have loved to have been here today celebrating. My brother, and of course my husband John, and my daughter Sasha. Of course, also none of this would be possible without a phenomenal research team. And I'm really blessed with some, as you can see, crazy, fun-loving research team members who really, again, a bit like my clinical team members, make every day coming into the office good fun. We have good fun both in the lab and outside the lab down on Hayling Island. And it's been brilliant working with them. And I also love working with my colleagues in the Institute, which is a really friendly, collegiate place to be and has really supported my career. And last but not least, as I always say to any aspiring, budding researchers, identifying a good mentor is almost more important than the project. And I have been really lucky enough when I came to St. George's to have met Tom. Tom's vision, his research integrity and his humility and the way he has supported me and countless others in their research careers, and his passion to really make a difference in the treatment of cryptococcal meningitis has been really inspiring. And therefore, I just leave you with this cartoon. We are here at St. George's, so this is St. George's, and I'm slaying the fungal dragon. I intend to continue to slay the fungal dragon until the last of us is reached. As Neil said in a recent lecture, the future is fungal, and I intend to keep on fighting fungus. And I just thank you for listening, and sorry that I went a little bit over.
Thank you, Tihana. It's a great pleasure to be able to thank you for your lecture, and thank you for giving us all, the insights into these amazing fungal infections, and telling us how they pose, such a dramatic challenge to us human beings in terms of the mortality and the morbidity. Sometimes underappreciated, as you say, by the general public, and also the fascinating biology of these organisms and how they pose unique problems when you think in terms of designing and testing new treatments. So thank you for that, and also, of course, your personal journey as well as your academic journey and the way those two have intermingled. So thank you for a fantastic lecture. Tihana will be willing and able to answer all questions in the short upcoming reception just around the corner from the lecture theatre. But I just wanted to spend a couple minutes just to share and celebrate with you some of the fantastic qualities that Tihana has as a researcher and as a colleague. And first off, if any of you are not familiar with St. George's, I think it's fair to say St. George's is a great place to do infectious diseases clinical work. If you're interested in infectious diseases, we have them all here in Tooting from all over the world, and every patient is completely different. And obviously, if somebody needs to come into the service here, they've been unlucky, but they've been relatively lucky if they come in under Tihana's watch, because she's a fantastic clinician with really great clinical judgment and 110% commitment to looking after her patients and getting them through their illness and getting the very best out of our NHS system to look after them. And of course, in the course of that clinical work, she's seen firsthand, we all see firsthand, the devastating effect that some of these fungal infections can have on immunocompromised and sometimes apparently normal patients. And as Tihana's shown you, she's taken that as her starting point and her inspiration to build the research program that she's given you a flavour of this evening. So she brings to her research critically that clinical insight, as well as huge originality and perseverance, because you need a bucket load of perseverance in this game. Thirdly, Tihana is a great team player. She's a great team leader, and she's an inspiration, as you've seen, to an increasing number of younger colleagues. And I think the younger colleagues are also lucky to have her as their mentor and supervisor going forward. And then just lastly to say, Tihana is fun to work with, as you've seen, and I'm just going to leave you with a couple more pictures of Tihana. On a personal note, I leave my office door open continuously in the hope that Tihana will stop by and we can have another chat, making plans and putting the world to rights. And she's a tremendous support to all the colleagues around her through the ups and, of course, also the downs of academic life. So thank you very much, Tihana. We are certainly very lucky to have you here at St. George's. And I think everyone is invited now to the reception just around the corner. So thank you again.